talk about the retina imaging technology that he was working on to help diagnose these patients even earlier so that the intervention can be earlier.
Certainly. Thank you for this opportunity. It's really wonderful to hear all these stories and certainly help us drive forward with this early diagnostic, which the pharmaceutical industries are, the clinical trials are failing just because there isn't early diagnostic which can start the treatment early or even start intervention that Derek mentioned, at least start lifestyle changes. So that's what we are working on, hoping that the retinal imaging can be useful in that regard. So briefly, I'm a associate professor in the Center for Drug Design. The center was founded by Professor Robert Wentz, and his invention for rational design of the first AIDS drug. That's the highest royalty earning invention of the university, and that's what helped us get into areas which are not just related to antivirals and help the project move forward. Really, Alzheimer's doesn't need an introduction. I just wanted to bring out two key players, proteins amyloid beta, which deposits in the brain as plaque, and tau, which is another protein also looked for intervention for developing new drugs. Our research has been focused on amyloid beta because we think that early diagnosis of these deposits before they deposit in the brain is what will drive the cure, hopefully the cure for the disease. So I won't go into the whole biochemical pathway, but you can see that this amyloid beta is a small peptide which eventually aggregates into the insoluble plaques and deposit in the brain. So if I would like to give an analogy for this whole process, a burning log analogy. If the amyloid beta, that is A beta, noted here, you can consider that as a fire. If our brain is a log and we set fire to the log, plaques are the ashes that are formed, aftermath. So the current diagnostics are based on detection of those plaques. So that's why the treatments are not useful. What we are aiming for is detection of these soluble species, which are present, which are toxic. So we have a compound which are targeted towards those soluble species I mentioned. And we tested that compound in a mouse model. And if this is a wild type mouse without any cognitive deficits, and Alzheimer's mouse with cognitive deficits, you can see the plaques deposited in the brain. When we treated these mice with the compound, it almost completely reduced the plaque formation in the brain. And when we looked at the memory of these mice, so we usually teach the mice, it's like a swimming pool. There is a platform which is hidden in the swimming pool. For 5 days, we teach them how to find the platform. And the control mice are able to find the platform, and the Alzheimer's mice are not. When we looked at the mice that were treated with our compound, they behave similar to the control mice. So we think that the compound really has a promise. It hopefully acts on the early stages of Alzheimer's, and we are currently going to approach FDA for at least undergo safety studies for this compound. Again, there are hurdles for going to an FDA, making larger amounts of these compounds to be tested in the clinic. So certainly funding in this area is necessary for that purpose. Again, pharmaceutical companies are not willing to take any risk for a new pathway, which they are not working on. So they are hesitant to invest their resources. So again, even if we have an early therapeutic, there is no use for the therapeutic if we can't diagnose it at earlier stage. As I mentioned, most of the diagnostic reagents are aimed at the plaques. They are radiation based, so you can't really administer them frequently. It has to be at least an annual exam. Expensive, not suitable for population screening. So we hope to develop a cost-effective and a non-invasive method. So we developed a spectral imaging method, which is really safe, no radiation involved. So initially we looked at just the brain tissue from normal and Alzheimer's patients. And we observed a spectral signature, that's what I would call it here. But again, imaging brain is not ideal. We needed something non-invasive. So we looked at the retinal tissue. Most of us believe that retina is a peripheral tissue, but it's not. When in the development stage, retina is a part of CNS, the nervous system. So whatever happens in the brain can be recapitulated in the retinal tissue. So that really encouraged us. We did our cell culture studies to prove that this really is reproducible. In the mouse model, we were able to detect the disease even before any cognitive symptoms are apparent. That encouraged us to move into a human camera. So we developed this camera and did a small scale study at the university. And did find that our technique was more sensitive towards the early stages that Derek mentioned, that it goes from mild, moderate, and severe. So at least in the university study, we were able to show that at the mild stage, our technique was sensitive. So last year in October, we licensed this technology to a company, Repispec, and who is currently undergoing clinical trials with this device. I would like to show a highlight of our research.
So you own the patent and you're licensing, is that correct?
The university owns the patent and it licenses it to a company.
$50 million people around the world are currently living with dementia, and Alzheimer's disease is the most common type.
But research suggests that a third of cases are preventable. Ellen Gallis shows us how an eye scan may be able to detect the disease early.
If the eyes are the window to the brain, then this camera could become a very powerful tool in detecting Alzheimer's disease. At the University of Minnesota, they've invented a camera that's helping them to see one of the signs of Alzheimer's earlier than ever.
It only takes a few seconds to do this exam.
The camera looks like something you'd see at the eye doctor. It shines a light into the retina and then measures the way the light is reflected. It detects a protein that looks like this, called amyloid beta. In early onset Alzheimer's patients, this protein uncoils and reflects light differently than a regular scan.
We shine different wavelengths of light into the retina.
By measuring the light levels, researchers can detect one of the first indicators of Alzheimer's. The camera is still only being used for research, but eventually, this technology could lead to much earlier detection and treatment.
Hopefully help develop drugs for early Alzheimer's, because the camera can also be used to follow the progress of a new drug as it's being, as the patients are being treated.
There are only 5 cameras like this one, and they're being used in different clinical trials all over the world right now, including Israel, Boston, the Mayo Clinic, and right here at the U of M. Reporting at the U of M, Ellen Gallis, 5 Eyewitness News.
And researchers at the U of M say they hope that one day that camera and preventative Alzheimer's detection will really become part of a routine eye exam.
So that was really encouraging, and we started in collaboration with the company 5 clinical sites. 3 of them are currently up and running. And I wanted to point out that there is a Mayo University collaborative grant which helped us start a site at the Mayo Clinic, and which is really helping us get some preliminary data. This is from that initial trial which just started a couple of months ago. And we are able to, so currently PET is the gold standard. And again, we are subjected to radiation. So these subjects are not showing any symptoms, pre-symptomatic. So we have 2 cohorts, PET positive and PET negative. With our technique, we are able to distinguish between these 2 groups. So we think that it can be a substitute. So at least to identify at-risk individuals, and maybe hopefully that will reduce the cost of the clinical trials for a therapeutic agent. So again, at this stage, our current measurement was at the mild stage. With the sensitivity of the technique, we hope that we can measure the preclinical changes before any symptoms start appearing. no complications of this method because we are just using the light source that's currently used in the eye clinic. This test is done in 10 minutes. And in mice, we have shown that we can even predict the outcome of a treatment. So we don't really have to conduct clinical trials for 5 years. In 1 or 2 years, we should be able to see whether the treatment is going to work or not. And we're currently studying whether this technique can be used for Parkinson's or Huntington's disease. So how can you really use it in a clinic? So we think that if someone is really interested, he or she can get an eye exam, get compared with the population of the same age group. If your results really stand out, then do the follow-up testing, reducing the whole cost for the management. Or you can, if you have predisposition, maybe you can start imaging early on. This can be incorporated in the annual eye exam, and maybe that will help diagnose this disease earlier.